of a whorl of stamens, representing the male reproductive organ, and the gynoecium represents the female reproductive organ. Well, in our previous module, we have discussed about the androecium, the male reproductive organs, which are stamens, as a part of the topic pre-fertilization, structures and events. Let us recall the structure of stamen. The stamen comprises of two parts, a long and slender stalk called filament and the terminal, generally a bilobed structure called the anther. Along with this, we also learned about the structure of pollen grains. Now, continuing the topic pre-fertilization, structures and events, in this module, we will discuss about gynoecium, the female reproductive organ. Well, the gynoecium represents the female reproductive part of the flower, that is, the pistil. The gynoecium may consist of a single pistil known as monocarpellary or may have more than one pistil known as multicarpellary. Generally, the term bicarpellary is used for two pistils and tricarpellary is used for three pistils. When there is more than one, the pistils may be fused together known as syncarpus or they may be free known as apocarpus. We know that the pistil is further divided into three parts. Yes, the pistil is divided into stigma, style and ovary. Now let us briefly discuss the functions of these parts. The stigma serves as a landing platform for pollen grains. It forms the pollen receptive tip, which is often sticky and can be of various shapes. The style is the elongated slender part beneath the stigma, which connects the stigma and the ovary. The basal bulged part of the pistil is the ovary. It is the part of the flower where the seed formation takes place. Inside the ovary is the ovarian cavity called locule. The placenta is also located inside the ovarian cavity. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1, as in wheat, paddy, and mango, to many, as in papaya, watermelon, or orchids. The female gamete of the plant, known as egg or ovum, is present inside the ovules. Now, let us accustom to the structure of a typical angiosperm ovule. The ovule being a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represents the junction between ovule and the funicle. A mature ovule consists of a food tissue covered by one or two future seed coats or protective coverings known as integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Opposite to the micropylar end is the chalaza, representing the basal part of the ovule. Here we use the term nucellus. Now you all might be wondering, what is it? Well, a mass of cells enclosed within the integuments is known as nucellus, and the cells of the nucellus have abundant reserve of food materials. Moving on to the next part of the ovule, we have the embryo sac. The embryo sac is a large oval cell in which fertilization and development occur. The embryo sac or female gametophyte is located in the nucellus. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. Before learning about this process, let us understand the term megaspore. Here, the megaspore, also called macrospores, is a type of spore that is present in heterosporous plants. The megaspore germinates into a female gametophyte which produces egg cells And the process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell, abbreviated as MMC, in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division, and meiosis results in the production of four megaspores. Here, I have a question for you all. Is the megaspore haploid or diploid? Think about it. Well, the answer to it is quite interesting. The megaspore mother cell or the megasporocyte is a diploid cell in plants which undergoes meiosis, resulting in the production of four haploid megaspores. And this also explains the importance of the megaspore mother cell undergoing meiosis. That is, the MMC or the megaspore mother cell undergoes meiosis in order to maintain the ploidy of the gamete. In a majority of flowering plants, the megaspore at the chalazal end remains functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte, that is, the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. And as discussed earlier, the embryo sac is a large oval cell in which fertilization and development occur. Now let us study about the formation of the embryo sac in a detailed manner. The functional chalazal megaspore enlarges to form the embryo sac, and its nucleus divides mitotically to form two nuclei, which move to the opposite poles, forming the two nucleate embryo sac. The nuclei in the two nucleate stage divide mitotically to form the four nucleate stage. This in turn forms the eight nucleate stage by dividing mitotically. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. Hence, we use the term 8 nucleate stage instead of the term 8 cell stage. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or the embryo sac. Observe the image shown here. 6 of the 8 nuclei give rise to 6 different cells when surrounded by cell walls. The remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus and constitute a single large cell known as the central cell. And there is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes to the synergid. 3 cells are at the chalazal end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8 nucleate, is 7-celled. Now let us move on to the next topic, that is pollination. We have learned that the male and the female gametes in the flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. So, how is this achieved? Well, here the process pollination comes into action. You might be well aware of this term, right? Yes, the transfer of pollen grains from the anther to the stigma of the pistil is termed as pollination. Also, flowering plants have evolved an amazing array of adaptations to achieve the process of pollination. They make use of external agents to achieve pollination. So students, in our next module, we will discuss about the process of pollination in detail and the external factors or agents responsible to bring about pollination.